Hello students, welcome to our online video tutorial class. So I think you have seen the previous videos, in which videos I have discussed about the structure of the flower as well as the structure of anther of this chapter. This is the second chapter, that is a sexual reproduction in flowering plant. So I have started with that specific portion, that is the structure of flower and structure of anther. So today I am starting with the next portion, that is the structure of pollen sac. You can see here in the structure of the pollen sac they have following the layers. Outer one is commonly known as epidermis. Next there is another layer that is known as endothecium. Third there is a layer that is a parenchymal cell layer. Next there is another layer that is tapetum, and the fifth one, another one, that is the microspore mother cell. So let's see whatever this specific layer signifies. First of all, epidermis, it is the outermost protective cell layer, and that is made up of epidermal cells. Keep it in your mind, epidermis is such kind of outer protective cell layer, and that is made up of epidermal cells. Next thing is that endothecium is the another layer which is actually present inside epidermis, and keep it in your mind, this endothecium is actually another protective layer also like the epidermis. And this endothecium is made up of endodermal cells. Like that epidermis is made up of epidermal cells, but endothecium is made up of endodermal cells. And these epidermal and endodermal cells which are present in case of root also, like those, this specific type of cells are also present in this layer. Next, parenchymal cell layer. What is that? Parenchymal cell layer is another layer where only the parenchyma tissue or parenchyma cells are only present, like the parenchyma cells are present in the leaf also. So those specific parenchyma tissue like cells, they are actually having this present in this specific cell layer. And keep it in your mind, due to presence of this parenchyma tissue, they are obviously containing the chloroplast, and for that reason they can perform the photosynthesis even, and they can produce the food also. Next, tapetum. Inside the parenchymal cell layer, if you enter, obviously you will see that there is another layer that is commonly known as tapetum. Keep it in your mind, this tapetum is made up of elongated cells. These specific cells are elongated shaped, and they are exclusively present there for providing the nourishment to the microspore mother cell. What is microspore mother cell? Microspore mother cell is a precursor of the microspore or the pollen grain. These specific type of cells are actually present centrally to the each and every pollen sac, and for providing the nourishment to the microspore mother cell, obviously tapetum is present there surrounding them, and this each and every cell of the tapetum is elongated obviously. And this microspore mother cell, they are start actually their division, their growth, and after when they will be just further getting mature, on that time they have formed the structure of the pollen grain present within the pollen sac. So this is the overall layers, or these are the overall structures present in case of pollen sac. Now we are just moving for the structure of the pollen grain, or rather we can say that is a microspore structure. So let's see. First of all, in case of microspore, we need to remember about their shape. The first thing is that they are actually showing oval or round or triangular shape. Most of the pollen grains are showing oval or round shape. Triangular shape is very rare, but it can be found for some of the flowers. Keep it in your mind, this pollen grain is commonly composed of following structures. Whatever the structures, first structure is exine, that is the outermost layer. Next there is another layer inside the exine, that is intine. Next there is a presence of vegetative cell layer or vegetative cell even. Next there is a presence of generative cell. These two specific cells are actually present in the cytoplasm of the pollen grain. Next there is a presence of callose wall. Next, germ pore and germ furrow. These two small as another one bigger pore like structure which are present on the surface of exine. So let's see about the structure of the entire pollen grain. First one, that is the exine, whatever I have told. Exine is the outermost protective covering. It is obviously present for the protection. Keep it in your mind, it can be spinous as well as it is containing the groove, or it can contain also furrows. So due to presence of this specific structure, spinous, groove, furrow, obviously that specific outer layer is actually getting protective. Next, keep it in your mind, this exine layer is commonly made up of a specific kind of protein that is known as sporopollenin, and due to presence of this specific type of sporopollenin protein, obviously the pollen grain is getting so much protective, because due to presence of the sporopollenin, the pollen grain entire structure or internal structures will be well protected. In the extreme environmental condition also, this sporopollenin is protecting the internal structures of the pollen grain. Next, exine layer, which is actually the outermost layer, that is divided into two layers. Already it is written here. One is sexine, that is the outer one, and another one is nexine, that is the inner one. Next, in case of some of the flowers pollen grain, their exine is secreting some sticky substances. Those are commonly known as pollen kit. So why this pollen kit is actually present? This pollen kit is actually present for getting attached with the insect body during the time of cross pollination. So that sticky substance, that is a pollen kit, is having in some of the pollen grains which are pollinated by the help of cross pollination. If you are entering within the next layer, you will be getting, that is intine. It is the inner layer of exine, and this specific layer, again it is subdivided into two layers like the exine, but specifically the names are not needed of these layers. Next, and the most important thing of the intine is this intine layer is responsible for forming the pollen tube, because during the time of the male gametophyte development, this specific pollen tube only, it is coming out from the intine, because intine layer is only protruding out and forming the club shaped pollen tube which is coming out through the exine. So intine is actually having that specific function to produce the pollen tube. Next slide, you can see at the first, that is a vegetative cell, that is a type of cell which is present in the cytoplasmic portion of the pollen grain, and this specific cell is compared to the another cell, it is larger in size. And another more important thing is that this specific cell is present for providing the nourishment to the male gametes. From where the male gametes have come, generative cell will be further dividing into two male gametes. So those male gametes are obtaining their nourishment from this vegetative cell. Next, we can see another cell, that is the generative cell, that is also present in the cytoplasm. But keep it in your mind, these specific cells are smaller in size and they are only producing the male gametes by the mitosis cell division. So when they are dividing mitotically, on that time only they are actually producing the male gametes. Callose wall, this is a specific type of wall which is actually separating the vegetative cell and generative cell from each other. Keep it in your mind, this specific wall is actually made up of complex sugar, that is callose, and that specific wall is only separating these two cells present in the cytoplasm. Next, there is presence of germ pore. What is about the germ pore? Germ pore is a small pore-like structure which is present on the surface of exine, and this pore, why it is present? Because of when the pollen grain will be landing over the surface of stigma, on that time that pollen grain is absorbing the nutrients from the surface of stigma. So those nutrients are actually entering within the pollen grain through this specific pore-like structure. Next, another porous structure is present, that is germ furrow, but keep it in your mind, these pores are compared to the germ pore, these pores are larger in size, okay, the diameter is more. And keep it in your mind, how many germ pores also will be present on the surface of exine, that much or that many germ furrow will not be there. Obviously the number of the germ furrow will be less than the germ pore. So germ furrow is actually present so that the pollen tube which is forming from the intine layer, that can come out through that specific structure, okay, through that specific structure means through the germ furrow. So these are the overall structural features whatever we have studied: exine, intine, then vegetative and generative cell present in the cytoplasm, and callose wall, germ pore and germ furrow. These are the overall structures present within the pollen grain. Going for the next topic, in the next topic it is written that we will be discussing about the microsporogenesis or formation of pollen grain. So first of all, we have to know what is microsporogenesis. Obviously the process of microspore or pollen grain formation from microspore mother cell, that is commonly known as microsporogenesis or pollen grain formation. So let's see. We can see here in this slide there are 4 different steps by which the microspore or pollen grain can be produced from the microspore mother cell. So let's see whatever the steps. First of all, we have written microspore mother cell which are having 2n number of chromosome containing, and this microspore mother cell divides first of all by the meiosis 1 cell division and then meiosis 2 cell division. When the microspore mother cell is dividing meiotically by the meiosis 1, obviously it will be forming microspore dyad. Dyad means 2, that means 2 microspore mother cells are actually joining with each other. Then when the microspore dyad has entered within the meiosis 2, then it will be forming the structure that is microspore tetrad. That means 4 microspore cells are actually present in a tetrahedral arrangement, and that specific structure is commonly called as microspore tetrad. Keep it in your mind, 4 microspores are there and they are actually having the tetrahedral arrangement. That's why this kind of name has been given. Now, from this microspore tetrad, when single microspores are getting separated from each and every one, obviously those microspores are considered as a functional microspore, because these specific microspores are getting activated. These microspores or pollen grains, now they have the capability to pollinate or to get transferred to the surface of stigma. So for that reason, when those microspores are present in the tetrad form, on that time they were not functional, but when they are getting separated from each other or they are getting dissociated, obviously they are getting functional, and each and every microspore or pollen grain will start functioning as a single unit for pollination. Going to the next slide. In this slide we can discuss about the development of the male gametophyte. What is about the development of the male gametophyte? First of all, the pollen grain has been formed by the process of microsporogenesis. But after formation of the pollen grain by the microsporogenesis, obviously that pollen grain has to be transferred on the surface of stigma, and when it is getting transferred on the surface of the stigma, after that from the intine layer of the pollen grain, the pollen tube is forming, and the formation of the pollen tube from the intine layer of the pollen grain, that is commonly called as male gametophyte development. So let's see whatever the developmental stages or developmental processes of male gametophyte. First of all, we can see here the pollen grain, it is first of all divides mitotically, and for that reason only the vegetative cell and generative cell, these two kind of cells are actually produced within the cytoplasm of the pollen grain. So we have discussed about the structure of the pollen grain. There we have seen what is vegetative cell, that is responsible for providing the nourishment, and generative cell, that is responsible for providing the responsible for producing the male gametes. But keep it in your mind, these vegetative and generative cells were not present from initial time. These two cells are actually produced by mitotic cell division from the pollen grain. Now these two cells, vegetative and generative cells, are getting separated by each other by the help of callose wall. Already that also we have studied. But when the developmental process will start, first of all this callose wall will get dissolved, because if the callose wall will not get dissolved, further the procedure cannot move forward. So for that reason the callose wall has got dissolved. Next, pollen grain in that condition containing vegetative and generative cell with dissolved callose wall, that is landing on the surface of stigma. Obviously if the pollen grain is getting accepted by the stigma, then pollen grain absorbs the sticky secretion from the surface of the stigma. That pollen grain absorbing the sticky secretion from the surface of stigma, just it will absorb intine layer of the pollen grain, that will comes out through the germ furrow, that also we know. And obviously for coming out through the germ furrow, that is actually forming the pollen tube. Now the point is, after formation of the pollen tube, the vegetative cell nucleus, that is entering within the pollen tube. First of all, out of the vegetative cell and generative cell, the vegetative cell nucleus only, it is entering within the pollen tube initially. Then generative cell nucleus dividing mitotically, because generative cell actually containing haploid number of chromosome, the nucleus are haploid number of chromosome containing. So they will divide mitotically, so that they will be producing 2 haploid male gametes. So just they will be making 2 haploid male gametes. Obviously those 2 male gametes now start entering within the pollen tube by following the vegetative cell nucleus. So first of all, when the vegetative and generative cell has got separated by the help of callose wall, after that when the pollen tube has been formed, then vegetative cell nucleus, it will actually enter within the pollen tube at first, and then by following the vegetative cell nucleus, the generative cell is also getting divided into 2 male gametes, and those male gametes will be entering within the pollen tube by following vegetative cell. Why the vegetative cell has entered at first? The reason behind it is the vegetative cells are exclusively present for providing the nourishment to the male gametes. So for that reason they have to move first within the pollen tube, then by following them, obviously the 2 male gametes will be entering within the pollen tube. So I think the developmental process of the male gametophyte we have all understood in a proper manner. Now we are just going for the next slide. Next slide is actually talking about the pollen uses or the importance, as well as whatever the diseases can happen from the pollen. So first thing is that we all know that pollens are rich in nutrients. They are containing so many type of proteins. So for that reason, obviously the pollens are rich in nutrients. From the pollens, nowadays the tablets are also made, pollen tablets, those are commonly told, and these pollen tablets are considered as a food supplement, because these are pollen tablets are containing a huge quantity of the protein. So when it is containing huge quantity of the protein, obviously it will be used, or it can be used as a food supplement. Next, pollen nowadays it can be used for producing the syrup also. This syrup is also acting as a food supplement. It is providing us more nourishment so that we will be getting more energy during our work. As well as in the last point it is written, you can see everyone, pollen is responsible for increasing energy in athletes and racehorses. So if some specific type of drink, energy drink, if anyone will have before running or before taking part within any kind of athletics, or in case of racehorse also it is applicable, that means that specific energy drink is containing pollen. Why the energy drink is containing the pollen? Because pollen is containing a huge quantity of the protein. So when the protein you are taking in a higher quantity, obviously that protein you will be providing us energy. When we will be doing any strenuous activity, obviously that specific protein food or the protein component which is present within the energy drink and which is obtained from the pollen, that will be broken down, and from there we will be getting the energy by the process of respiration. Next it is written, the whatever the type of diseases also can happen from the pollen. From the pollen, obviously some people have the allergy, so pollen allergy it can happen, as well as from there the bronchial infection, that means the infection in the primary and secondary bronchi which is present nearby or within the lung. So those specific bronchial structure can be get infected the inner wall due to the pollen, because pollen is actually considered as an allergen. So obviously when they are considered as an allergen, obviously they are causing allergy. Next, chronic respiratory disorder, that means if anyone has the pollen allergy, they can have such kind of respiratory disorders also, and that is chronic. Okay, for a long time, from the birth time they are having this kind of disorder, like that asthma, bronchitis, this kind of diseases also can have within any individual for the pollen grain from the time of birth. So these are the diseases which are actually occurring for the pollen grains. Okay, going for the next slide. In the next slide you can see properly about the viability of the pollen grain. Viability of the pollen grain means we are talking about the capability of the pollen grain up to which specific time that specific pollen grain remain capable or remain viable, so that it will pollinate any other specific flower. So that specific phenomena, that specific type of property of the pollen grain is known as viability. That means up to how much time that pollen grain remain active, so that it can pollinate any other flower and it can help in the pollination process rather. So pollen viability, it is always depending on the temperature and humidity. Keep it in your mind, if in any of the environment the temperature is very high or temperature is very low, as well as if humidity is very high or humidity is not even present, the weather is very dry, on that time obviously the pollen viability should not be maintained in a proper manner. So keep it in your mind, the temperature and humidity, these two factors are very much necessary, and optimum temperature and optimum humidity, that is the intermediate temperature and intermediate humidity, will be always required for maintaining the activity or viability of the pollen grain. Here some examples have been given also. In case of wheat and rice, this kind of cereals, the pollen viability most of the time it is present 30 minutes. In case of Rosaceae, Leguminosae and Solanaceae plants, this pollen remain viable for few months, as well as pollen can be preserved within liquid nitrogen in the temperature of minus 196 degrees Celsius. So that is commonly known as cryopreservation. So that means pollen can be cryopreserved also for maintaining its viability. Maybe it is kept within the liquid nitrogen in minus 196 degrees Celsius temperature, but keep it in your mind, they can be frozen, but they cannot be get inactivated. They remain active also after so many years. So for that reason this cryopreservation process is so much necessary. Okay, so this is the pollen viability. It can vary from plant to plant obviously, and the viability is always depending on the temperature as well as humidity. Keep it in your mind. Next we are going for the structure of pistil. Structure of the pistil, we have already discussed about the structure of androecium in the previous of this video. So here we will start discussing about the structure of pistil. So let's see. First of all, we can see here the pistil is commonly made up of following structures. Whatever the structure, that is the first one, that is the stigma, next one, that is the style, and the third or the lower portion, that is ovary. So please keep it in your mind that first of all, what is stigma? Stigma is the upper stage like structure. Always it is present towards the upper portion of the pistil or carpel. So it is obviously a stage like structure. Why? Because it is actually responsible for landing the pollen grain on it. So for that reason they are actually stage like structure or platform like structure showing. This space for obviously for the pollen landing already I have told, and keep it in your mind on the surface of the stigma the sticky secretion is always present so that the pollen grain which is coming on the surface that can adhere properly. Next you can see that is a style. Style is the filament like structure. This is a filament like structure. In the figure you can see just under the stigma, this is a filament through which actually the pollen tube can run or it can proceed during the time of pollination. So for that reason style is present. As well as style, another function is to hold the stigma portion. That is also another function. And the lowermost portion, this is the ovary. Keep it in your mind, this ovary is forming the fruit in future, and keep it in your mind within the ovary there is a partition, and that partition or the membrane like structure which is present, that is commonly known as placenta. Keep it in your mind, so many ovules, that means the units of ovary, those ovules are remain attached with the placenta, and these ovules in future only producing the seeds, okay, within the fruit. And keep it in your mind, each and every ovule is attached to the placenta by the help of a stalk like structure that is commonly known as funicle or funicle. So you can see here in this diagram also, this is the entire ovary that is subdivided into two chambers, and within the chamber obviously these are the ovules and they are actually arranged in a flower like manner, okay, and in middle there is a partition or wall. That specific partition or wall is called as placenta, from where the ovules are remain actually attached by the help of funicle or funicle. So this is the overall structure of the pistil or carpel. Going for the next slide. In the next slide you can see about the types of ovary based on the number of chambers. Whatever the types of ovary based on the number of chambers, let's see. First of all, here the specific word it is used, that is locule. Locule means actually the chamber. So based on the number we have segregated the entire ovary into multiple types. So if that ovary is containing single chamber only, that will be considered as a unilocular ovary. That means there is no placenta, that's why only it is unilocular. If there are two locules or two chambers are present, that means single placenta is present. And if there are three locules or three chambers are present, that means it is a trilocular. If there are more than three locules, that means four locules are present or chambers are present, obviously they are known as tetralocular. If more than four locules or chambers are present, then it is known as pentalocular. And if more than 5 chambers are present in that ovary, on that time that specific ovary is known as polylocular. That means based on the number of locule or chamber only, we have classified the ovary into these 6 different types. Going for the next slide. In the next slide you can see types of flower based on the number of carpels. So here we will be discussing about the carpel based on their association or based on their number, how many types of flower we can see. First of all, here also, if single carpel is present, that means in case of that flower obviously that will be called as monocarpellary. Bicarpellary flower means they are having 2 carpels. Tricarpellary flower means they are having 3 carpels. Tetracarpellary flower means they are having 4 carpels. Pentacarpellary flower means they are having 5 carpels, and polycarpellary flower means they are having more than 5 carpels. So that means based on the number of carpels also we have differentiated the flower into these 6 different categories: monocarpellary, bicarpellary, tricarpellary, tetracarpellary, pentacarpellary and polycarpellary. Going for the next, that is the types of flower based on the fusion of carpels. Now, that means now we have discussed about the number of carpels, we have got the type of flowers. Now based on the fusion of carpels, how many flowers we can see or how many types of flowers we can see. Mainly there are 2 types of flowers can be found. One is syncarpus, another one is apocarpus. Keep it in your mind, apocarpus flower is a type of flower where the carpels are all free. They are not united even, they are not connected even with each other. So they will be known as apocarpus flower. And another type of the flower that is where the carpels are united with each other and they are forming a single unit. So those kind of flowers are commonly known as syncarpus flower. So fusion of the carpels in the flower. So obviously they will be known as syncarpus flower. So I think up to this specific slide we have understood all the points whatever we have discussed. We have started from the structure of pollen sac. We have discussed here about the entire layers of the pollen sac: epidermis, endothecium, parenchymal cell layer, tapetum, microspore, mother cell, everything. Then we have discussed about the pollen grain. In case of that, outer layer is exine, then intine is present, and inside the cytoplasm there are two type of cells, vegetative cell and generative cell, which are separated from each other by the help of callose wall. Then there is a presence of germ pore and germ furrow on the surface of exine. Then we have discussed about the microsporogenesis, that means the formation of the pollen grain. Then we have discussed about the how the pollen grain, when it is landing on the surface of stigma, from their pollen tube is forming, and how the various cell like structures are coming within the pollen tube, that we have discussed in this specific point in these two slides. Next we have discussed about the uses of the pollen grain as well as whatever the type of diseases can happen from the pollen grain. Next we have discussed how much time the pollen remain viable, and that is obviously depending on some of the factors like temperature and humidity. In case of some plants, how much pollen viability can be found, that also we have discussed, and cryopreservation, how it is helping for maintaining the viability, that also we have discussed. Next we have discussed about structure of pistil. There are three portions: stigma, style and ovary. Whatever their basic structures, structural configuration, that we have discussed. Next we have discussed about the types of ovary based on the number of chamber. So you have seen here unilocular, bilocular, trilocular, tetralocular, pentalocular and polylocular type. After that, type of flowers we have seen based on the number of carpels, obviously monocarpellary, bicarpellary, tricarpellary, tetracarpellary, pentacarpellary and polycarpellary flowers. And at last we have discussed about the types of flower based on the type of fusion. If the carpels are fused, then it is known as syncarpus flower. If the carpels are not fused, in case of any of the flower, those are known as apocarpus flower. So today we are finishing our video up to this slide. In the next video we will be discussing or start discussing from the point that is the structure of the ovule and so on. So I think you have understood the entire video and you have enjoyed this video a lot. So thank you everyone.